We're going to get back going again, and we're going to move the focus just a little bit down to the end of the body, to the diabetic foot, and to the problems of preventing limb loss and promoting wound healing in patients with diabetes and vascular disease. But to take the specific focus on how dysmetabolism or how changes in metabolism might be targets to improve limb care and wound healing, I've asked a couple of people to speak to this afternoon, and we're going to kick off with a recorded talk by Kathy Gallagher. Kathy is probably well known to many of you as a really leading researcher in diabetes wound healing and macrophage function. She is a surgeon scientist, professor at the University of Michigan of surgery, microbiology, and immunology, which reflects the intersections of her research. And so she's going to set the stage for us with a little bit of reflection on some of the molecular factors that control wound healing beyond blood flow alone. So let's play her video.
Thank you for attending this talk, and I'm sorry I can't be there in person. I'm going to focus on epigenetics in diabetic wound healing with sort of a cardiometabolic focus, as diabetes is clearly a cardiometabolic disease with a lot of implications in the vascular surgery space. So I'm just going to kind of do a brief overview about wound healing, what goes awry in diabetic wound healing, some of the roles of various immune cells and structural cells, and then sort of what our lab has looked at as it relates to epigenetics. So normally following injury, you have a very quick response with platelets and fibrin forming to kind of cause quick hemostasis and sort of stop the bleeding. You have various immune cells that start to appear here. You have a coagulation cascade that gets activated. But again, platelets kind of being the main player here. This is followed by an inflammatory phase within an hour of injury to 48 to 72 hours, where you have more inflammatory cells, in particular neutrophils and macrophages. The macrophages are initially existing in a pro-inflammatory state, where then they switch to a reparative state. They secrete multiple pro-inflammatory cytokines. They participate in phagocytosis. Additional leukocytes come. This is where you have MMPs, elastase, and other things. This is then followed by macrophage switch to a more reparative type phenotype, where you have TGF-beta, VEGF, HIF-1 alpha. Fibroblasts begin to come in. You have granulation tissue and angiogenesis in this proliferative phase, which is 48 hours to 2 weeks later. The fibroblasts start to produce collagen. Again, you have more VEGF and HIF-1 alpha in this phase. And then the granulation tissue eventually fills the wound. This is followed by the remodeling phase, where the type 3 collagen gets replaced by type 1 collagen, and some cross-linking occurs. So in normal wound healing, this is a continuum. It kind of happens through these different phases where these different cell types play key roles. But again, it kind of moves along this continuum. These are kind of the four main phases that I just talked about. My lab particularly focuses on this inflammatory phase and looking at the macrophages and sort of their transition, although in the setting of diabetes, there have been multiple cell types, particularly fibroblasts, keratinocytes, endothelial cells that are involved in these proliferative and remodeling phases that have also been shown to be epigenetically altered and to be altered in the state of disease. But we know that in diabetes, you have this proliferation, or this inflammatory phase, where you don't seem to have these macrophages transition from the inflammatory phase to the reparative phase. And so our group has really kind of looked hard at why that would be. And a lot of our initial work was based on this concept of macrophage plasticity, and why do macrophages not transition from this inflammatory to this anti-inflammatory phase and allow the wound to kind of progress through this normal cascade. So we know type 2 diabetes, again, kind of a cardiometabolic disease, influenced a lot by diet, environment, various environmental factors. Epigenetic modifications influence gene expression, usually based on environmental factors. They're non-genetic based changes. Lots of different types of epigenetic changes that have been studied: histone methylation, or histone modifications, DNA methylation, microRNAs. Again, we really are going to focus on histone modifications. That's what our lab tends tends to focus on. But how is this epigenetic signature induced in macrophages in the setting of wounds? Again, we kind of look at these histone tails, which are shown here. There's various histone modification enzymes or chromatin modifying enzymes. We're just focusing on the methyltransferases here. There's also demethylases, there's succinolases, there's all different types of enzymes, but basically they all function similar in that based on the residue that they either add some moiety to, in this case a methyl group, they open the chromatin, allow transcription factors to come in and bind and effectively turn gene expression on. For the case of these demethylases, for example, if they bind at a particular residue, they can close the chromatin, you don't have binding, and you effectively turn genes off. So it's a way of modulating gene expression, which effectively controls the cell phenotype in the setting of the wound. So why do we care about this? Well, obviously in wound repair or tissue regeneration, it's really important that these macrophages and other cells have cell plasticity. In the setting of diabetes, it's a very costly and very significant problem for all of medicine, and particularly the vascular space. It's the leading cause of lower extremity amputation in the United States, so a really costly problem, and we really haven't had effective treatments to sort of fix this or to be able to repair sort of the problems that we see with these diabetic wounds. Now, again, we're really focused on, one problem, which is the immune system dysregulation that occurs in diabetes, but it's very well known that there's also impaired angiogenesis, impaired blood vessel issues, peripheral neuropathy. So again, the problems are multifactorial, and there's lots of people kind of focusing on these different areas. I'm just really choosing to hone in on this one particular area where I'm looking at immune cell function and why this has gone awry in the setting of diabetic wounds. So again, majority of the monocyte macrophages that you see in wound healing have come from monocytes in the blood, and prior to this, they've come from the bone marrow. They initially are in an inflammatory state. They shift to an anti-inflammatory state to allow the wound to progress through these 4 phases. This doesn't happen in diabetes, and so we're really looking at why do these macrophages not flip in the setting of diabetes. I'm going to go over very quickly a couple of different epigenetic enzymes that we've looked at and published on, and kind of just tell you a little bit about them and how they might control macrophage phenotypes. Some of these are relevant in other cell types as well. The first one we studied was called SETDB2. It's a methyltransferase. It effectively turns genes off. And I'll skip right to the punch. It turns inflammatory genes off. So it's really important, and we identified it first in normal wounds as going up in macrophages at the time when you're trying to switch from that inflammatory to that proliferative phase. And so SETDB2 gets increased via an interferon JAK-STAT mechanism, essentially comes in, turns inflammatory genes off, and then the macrophage switches to a more reparative type macrophage. What we found that in diabetic setting, both murine and human, SETDB2 does not get increased, so you don't get that inflammatory break, and then you get, unfortunately, dysregulated inflammation that occurs from the macrophages. So this was kind of just one look at that. Additionally, and as is true for a lot of these epigenetic enzymes, this also controls xanthine oxidase and uric acid metabolism. So we also found that SETDB2 is kind of interlinked with inflammatory uric acid metabolism. And so you also, by not being able to upregulate SETDB2, you also can't turn off your uric acid, which is a big problem in diabetes in general, but also in diabetic wound healing. So again, our group is looking at ways to increase SETDB2 via modulation of this interferon JAK-STAT cascade. Another enzyme, and this is just kind of showing what we've talked about as kind of ways to inhibit that pathway. Another enzyme that we've looked very specifically at is a demethylase. It's known as JMJD3. It essentially removes methyl groups and turns gene expression on. So it's basically going to be an activator. And we found that JMJD3 was significantly increased in diabetic wound macrophages, again, from human and murine models of diabetes, seems to control kind of some classic inflammatory genes, TNF, IL-1B. When we look at it with our floxed mice and such, we can see that it's regulating these genes. In humans, we can see that in type 2 diabetic wounds, JMJD3 is really strongly increased. So again, and it seems to be driven by an IL-6 receptor-mediated mechanism. So we were looking at ways to decrease JMJD3. It's also regulated via IL-6 and JAK-STAT3, both in human macrophages and in murine macrophages. But we were looking specifically, and this is kind of a new angle that our lab has taken over the last 5 to 10 years, where we're using drugs, nanocarriers, that specifically target the macrophages and can bring some of these epigenetic enzyme inhibitors, and in this case, the JMJD3 inhibitor, to the macrophages in the wound and reverse the phenotype. So by decreasing JMJD3 with this inhibitor in the macrophages specifically, we can cause them to move toward a more reparative phenotype and less inflammatory phenotype. So we decrease the inflammation here. So this is just one example of using cell-targeted nanotherapy with these epigenetic enzymes to sort of target these pathways. A newer story that we've been looking at, and we know that it's very important that you have myofibroblasts and that macrophages, we know, transition to myofibroblasts. Another epigenetic enzyme that plays a role here is called WISC1. It's kind of a newer enzyme that we were looking at, which increases gene expression. So again, important to transition to these myofibroblasts, which help promote wound repair in that proliferative and remodeling phases. We know that diabetic wounds don't have enough myofibroblasts, and that's one of the problems. So again, we've looked at how this enzyme WISC1 allows for upregulation of the myofibroblast genes and effectively allows for this cell transition. It's modulated via this TGF-beta mechanism. This is just kind of some of the background of how we identified that WISC1 was going up in normal wounds, and we don't see it, unfortunately, in diabetic wounds. And so we're starting to look at ways that we can modulate this WISC1, again, using some of the same nanotherapy in order to try to inhibit this WISC1 and have regulation and increase the number of myofibroblasts that we can see. This is just showing in human single-cell sequencing. We also see this in our spatial sequencing, that you can see the WISC1 is significantly decreased in the diabetic wounds, and we want to be able to increase WISC1. It turns out it interacts with REL-A and a common NF-kappa B transcription factor, and so by targeting REL-A, we may be able to upregulate the WISC1 and allow for transition to myofibroblasts, as you can see here with the spatial sequencing. So just kind of a quick summary of mechanism. So I just wanted to show you a few of those stories, but this is why, why we're looking at this. And again, there's lots of different avenues to look at this to try to promote regulated inflammation, to promote sort of progression through this normal wound healing cascade in the setting of diabetes. Thank you.
So Kathy's not available to take questions, but we'll be connecting with her after the meeting for her inputs on our sessions. But I think a perfect follow-up to that is going to be Kara Spiller. Kara is a professor of biomedical engineering at Drexel. She's part of the NIDDK Diabetic Foot Consortium, which is how I've gotten to know her and appreciate her science. And I think you'll be hearing from her perspective a little bit more maybe on the clinical side of some of the immunology of wound healing.
Great. Well, thank you so much, Dr. Conte, for inviting me today. It's really an honor to be invited to speak amongst this elite group. And I'm also feeling a little intimidated because I am not a clinician. I'm an engineer, and but I'm in biomedical engineering. And so, we biomedical engineers really rely on collaborations with clinicians in order to make an impact for patients, because that's not something we're able to do directly. So really looking forward to meeting you all and continuing to speak with you. So in my lab, we focus on engineering macrophages, the primary cell of the innate immune system. So Dr. Gallagher's talk was an excellent primer. And I just wanted to say before I started that I just started my first company last week, which I'm very excited about. This was thank you. This was a professional bucket list sort of thing for me because, as I said, since I'm not a physician, really the only way that I can make an impact for patients is by starting companies to try to bring my inventions to them, and that necessarily requires working with people like you. So, but it's a significant conflict of interest, so that's an important disclosure there. It's only the company that I founded is going to be licensing the macrophage cell therapy stuff that I'll talk about at the first half, and then in the second half I'll be talking about biomarkers and personalized medicine based on macrophages. Okay, so before I get into it, I just wanted to explain that I would say my mission is to increase accessibility to it for advanced therapeutics. And this is because most drugs are not wonder drugs, right? I think we could argue that the GLP-1s are probably getting as close as we've had to wonder drugs. But for many of our sickest patients, we're probably going to need advanced therapeutics that are really more complex than the healthcare system is designed to afford right now for the volume of patients that you all see. So while there's a lot of people taking different approaches to increase accessibility of these advanced therapeutics, I'm taking an approach from what my training was, which was engineering innovations. So first of all, I focus on macrophages in my lab because they're major regulators of tissue repair, and they control all of the other cells involved in tissue repair in every tissue in your body. So this is just some schematics of the tissues that I focus on in my lab, but macrophages are actually critical for wound healing in literally every tissue in your body. And when macrophages become dysfunctional, that's when you see impaired wound healing, as Kathy just described. So just to recap the main points of what she has been a huge leader in showing is that macrophage phenotype is critical for healthy tissue repair. And at early stages of tissue repair, macrophages must be pro-inflammatory. And then at later stages, they must transition to reparative phenotypes in order for proper tissue repair to unfold. And a lot of times you hear people speaking primarily about that reparative phenotype or the anti-inflammatory phenotype, and that is very important, but now we know that the inflammatory phenotype is also really important for setting the stage for wound healing and angiogenesis in particular. And in particular in the area of vascular ulcers or venous leg ulcers, a lot more research is coming out showing that pro-inflammatory treatments can actually be helpful in these wounds. And I think that comes back to a lot of the basic science that has been done about the role of pro-inflammatory macrophages in angiogenesis in particular. If you've ever heard of the M1 and M2 paradigm of macrophage phenotype, that refers to pro-inflammatory M1, reparative M2, but we try not to say that anymore because now we know that there's so many different types of phenotypes that if you say M1, M2, the brain wants to think there's only two types of phenotypes. It's just like impossible to wrap your head around that if you say M1, M2, so we try not to say that anymore. And then I'm not going to talk about the metabolism, but it is now known from Dr. Gallagher's research and many other leaders that these phenotypes are metabolically regulated upstream of a lot of other signals. And so in metabolic syndromes, of course, you're going to have defective macrophage behavior, and that's going to lead to defective wound healing. Okay, so this is probably a poorly laid out slide to make the point that I wanted to make on this slide, which is that I have spent a lot of time learning how to culture macrophages from humans in vitro and characterize their interactions with other cell types. I should have done a better slide to say that. I'm saying it now because as we think about ways that we can collaborate, and I'm very eager to collaborate with all of you, one piece of expertise that we have in my lab is culture of human immune cells in complex 3D systems with other cells involved in healing. And so now, after today's talks, I'm dying to give GLP-1 drugs to macrophages in vitro and then take those macrophages and expose them to endothelial cells, and that's probably the link by which GLP-1 receptor agonists influence endothelial cells is via macrophages. And we can test that pretty readily. But the other point I wanted to make here is that there's 4 different phenotypes of macrophages depicted on this slide. And in vitro, it's pretty straightforward to push macrophages to different phenotypes and study what happens. And through that, we've found that different phenotypes of macrophages play different important roles in wound healing. But controlling them in vivo is a lot harder. So I launched my academic career. I've been a professor at Drexel since 2013, so I built a career on making biomaterials that could control macrophages in situ through drug delivery or protein delivery. And I got a lot of papers out of it, and that's what I needed to do to get all my promotions and everything. But at some point, I got promoted to my final stage, and I thought, is this all there is? All I'm going to do is publish papers and beg the study sections for grants for the rest of my life, And so I really started thinking more translationally, like how could I get back to what got me into science, which was wanting to help people who are in need of help, I mean, that's how I was raised, and because I didn't go to medical school, I wanted to get back to that. And so I started thinking that maybe it's naive to think that by simply releasing a drug or a protein from a biomaterial in the setting of a wound, that's going to be sufficient to overcome the microenvironment that is really controlling macrophage behaviors. Or maybe it's naive to think that these patients who have had decades of epigenetic modifications to their immune cells residing in the bone marrow, really, I'm going to override that with a simple biomaterial releasing one or two drugs, right? And in fact, there's a lot of excellent research showing from many people in this room showing that in very sick patients or very bad wounds, the macrophages are either too few or too defective. And maybe that's the reason why all these treatments that have been developed that maybe worked in vitro or in mice aren't going to cut it in humans, right? So that got me thinking about macrophage cell therapy, because it's much, much easier to control macrophages ex vivo. Well, I mean, really, it's probably easier to control them in vitro and then transplant them into the wound rather than trying to override the signals at the site. But the problem with cell therapy is they're really expensive right now. So the most famous cell therapy that you may have heard of are CAR T cells. And these were first approved in 2016, 2017, and they've revolutionized medicine by curing previously incurable diseases, particularly hematologic malignancies. But they all work by genetic engineering of the cell. So you take the cell from a patient, you genetically engineer them in the lab, and then you transplant them back into the patient. And the genetic engineering part is the reagents that go into that are themselves difficult to manufacture. So usually it's viral vectors or other DNA delivery systems. They're hard to manufacture. And so then that raises the price of the final product. The other side of the cost is the logistics of taking a patient's cells and engineering them in a supply chain situation. And so I've got some ideas about how to solve those problems as well. But the lower hanging fruit was to fix that, is to not use genetic engineering to control the cells. And for macrophages, you shouldn't have to use genetic engineering because they're so exquisitely sensitive and they respond so readily to biomaterials that can be manufactured in large batches very inexpensively. So basically 10 years ago, we set out with an engineering question, which was, could we design a way to control macrophages for cell therapy that circumvents the manufacturing and supply chain issues associated with conventional immune cell therapies? And so the challenges that needed to be overcome was, first of all, macrophages, because they can quickly shift their phenotype so dramatically, super quickly. If you give stimuli to a macrophage to push them into a certain state and then transplant them into a wound, they will be completely different within a day. They do not maintain their phenotypes. There has to be a way to lock them in a phenotype. But as I said, gene delivery methods are very expensive. They don't work that well, and also they're pro-inflammatory to macrophages. But also we have to address the manufacturing challenges up front, because otherwise we're never going to make products that make it to patients. So 11 years later, here's what we came up with, which was feeding macrophages biodegradable polymeric microparticles ex vivo, and those microparticles slowly degrade inside the cells and release the drugs from the inside out, thus controlling their phenotype without the need for any genetic engineering or anything. And this would, because the microparticles are already used in FDA applications and can be manufactured at scale, it would be a lot less expensive than what's currently available. So this was the idea. Nothing like this had ever been done before. So the first question was, will they even keep them inside them for a long enough period of time for this to work? Because that was not a given. Maybe they would spit them back out. Who knows, right? So we spent a while studying that. So what you can see in this video are red microparticles inside green macrophages, and the green macrophages are crawling around in vitro and interacting with other cells. And we found that in vitro, they do keep the microparticles inside them for several weeks, which was good. And it did control their phenotype in vitro for several weeks. So the drug that we ended up going with for a lot of our applications is dexamethasone. It was selected because it's potently anti-inflammatory and it acts on intracellular receptors. So if we were going to use something that controls macrophages from the inside out, it had to act inside the cells, as opposed to a lot of other drugs act on the outside of the cell, so that wouldn't work. So we call these cells DexMPMAX, macrophages that have been fed dexamethasone microparticles. So in a couple published papers, we've shown that even if you put a lot of pro-inflammatory signals on the macrophages, if they had been fed, if they contain these microparticles inside them, it dramatically suppresses pro-inflammatory signaling. So it does override the extracellular pro-inflammatory cues. And then as a proof of concept that this would also work in vivo, we implanted them in mice in a subcutaneous implantation. And what I'm not showing here is that they do retain their microparticles inside them, and it did suppress pro-inflammatory signaling, but we also noticed that it promoted reparative phenotype activation. And that marker right there, CD163, is particularly associated with angiogenesis, or angiogenic behavior of macrophages. So we tested if these cells could improve angiogenesis in this model, which is actually, it's kind of a weird model because it's a subcutaneous implantation, but it's considered a rigorous model of angiogenesis because there's so few blood vessels in the backs of these mice that I've never seen anything cause vascularization into the centers of these thick scaffolds except for these cells. So far into the center of these thick scaffolds, we see mature blood vessels going all the way in within 2 weeks. And free dexamethasone microparticles do not have this effect. So so we have a couple controls here. So the vehicle is just a scaffold. You have unmodified macrophages, they don't help. Macrophages loaded with blank microparticles, they don't help. Here's the macrophages loaded with the dex microparticles. They improve the number of endothelial cells, and I think it's hard to see, but they're the only one where blood vessels actually make it into the center of the scaffolds. And then free dex microparticles have no effect. So they really need to be engineered ex vivo to see this effect. And then we started characterizing them more. We were like, well, that was cool. Let's keep learning more about how this works. Even just from like a basic biomaterial science standpoint, there was a lot of interesting questions that emerged. And one of them was, can they also suppress profibrotic signaling, because macrophages can play a major role in fibrosis, and so if it also suppresses that could be useful. And they did. So one of the major proteins that are secreted by macrophages in fibrosis is actually not TGF-beta, but osteopontin, encoded by the SPP1 gene. And here you can see that macrophages that contained the microparticles dramatically suppressed osteopontin signaling almost as much as macrophages that were in continuous dexamethasone the whole time. And then, but the macrophages are loaded with polymers. So I thought, well, maybe they'll be defective in phagocytosis or collagen degradation because they're loaded up with polymers. That would be a problem if I wanted them to degrade, if I wanted them to do their job in vivo, right? So we did a collagen degradation assay and found unexpectedly enhanced ability to degrade collagen in vitro. This was weird because dexamethasone, which is on here, so soluble dex-treated macrophages, they do not increase their ability to degrade collagen. And macrophages loaded with blank microparticles do not increase their ability to degrade collagen. But when you put them together, they do. So a theme that came up a few times today was, so I think Dr. Beckman said some of the best drugs were invented for something else, and then you realize they were good for something, and then you have to go back and figure what that is. So that's where we're at right now. We're trying to figure out why this happened. Here's my other plug for metabolism is I've got some preliminary data, but it wasn't quite ready for prime time, that we think the reason why they're degrading collagen may be that they're utilizing collagen as a fuel source because of altered metabolism by the dexamethasone, but that's kind of a unproven and kind of wild theory. But I just wanted to pivot for a second to pulmonary fibrosis to show you that we proved that also happens in vivo, because it's a new way of approaching fibrosis to deliver cells that could actually clear fibrotic tissue. So we got this animal model up and running called the repetitive low-dose bleomycin model. So if you give 1 dose of bleomycin to mice's lungs, fibrosis develops, but then it spontaneously resolves. And so you can't test something that you think is actually going to clear fibrosis in that model, right? It's like, and none of those drugs that work in that model translate to humans. So that wasn't going to work to test our theory that the macrophages could actually degrade fibrotic tissue in vivo. So this model, what you do is you give them 3 doses of bleomycin every 2 weeks for 4 weeks. Then you wait 6 weeks so that by week 10, the fibrosis in the lungs is established and it cannot spontaneously resolve. And that's the time point at which we administered our engineered cells to see if they could actually clear fibrosis in this setting. And we found that within 7 days, there was a greater than 50% clearance in fibrotic tissue after just a single dose of the macrophages. And that effect lasted to day 28. That's the latest, maybe longer. It didn't come back at least until day 28. And we measured it with 3 independent methods at this point. So whole lung micro-CT, a hydroxyproline assay. This is like a biochemical assay of fibrosis, and then histology, which I'm seeing here. And to my knowledge, no one else has ever reported true clearance of established fibrosis. And so that's why I started the company based on this data. And so now we're trying to position this for idiopathic pulmonary fibrosis, which is really a terrible disease. But I wanted to share it with you all because I know that fibrosis is also relevant to this community. I just thought I'd also share some interesting things about the other controls that we included, which was, so the same controls we had before, which was unmodified macrophages, blank microparticle macrophages, and then free DEX microparticles and soluble DEX macrophages. None of these have any effect on fibrosis, and neither do any of the drugs that are clinically approved for treatment of IPF, which we know that in IPF, the fibrosis just gets worse and worse until you die. And so, yeah, so I am still pursuing that, but looking forward to your feedback on how this might be useful in applications that you're interested in. But I think Dr. Conte actually invited me to talk about my personalized medicine work, and so I want to spend 5 minutes, if I can, talking about this, which is another approach to increasing accessibility of advanced therapeutics, which is that, so I'm working on the actual therapeutic side, so using streamlined manufacturing. I'm trying to get a cheaper cell source so that it's as cheap as possible. But what if we could tell who was not going to heal in response to less expensive treatments? And this is sort of like a uniquely American problem where we need to explain to the insurance companies or to Medicare why we should pay for advanced treatments for these patients. You know, even though your clinical expertise, you probably can tell this patient has high risk written all over them, but still you have to try 4 weeks of standard of care or something. Now I'm thinking specifically about diabetic foot ulcers. And so it's actually been known for a long time that there is a need for biomarkers in diabetic foot ulcer care, because the problem is that many patients, so 50% of patients, give or take, will respond to moist gauze, essentially, as the treatment. And that treatment is so inexpensive. But if they don't, they should be getting more advanced therapeutic, which will increase the likelihood of healing. But it's impossible to tell who's going to heal and who's going to not heal in response to those lower cost treatments. And there's been a lot of excellent papers published showing that, like, right now they use wound size, just length times width measurement of the wound. And if it changes over 4 weeks, if it decreases over by 4 weeks, that's only 58% accurate for predicting healing. So if it doesn't decrease, it's pretty good at predicting non-healing. But if it does decrease, it's a coin flip. It might still not be healing. So a long time ago, we set out to see if markers of macrophage phenotype could be used to predict who was going to heal or not. So in one of the papers that we published on this 10 years ago, we showed that not a single gene related to macrophage phenotype could differentiate between healing and non-healing ulcers. So these graphs are intended to show no difference between healing and non-healing over time. But we thought, but patients are so different from each other. Maybe it's crazy to think that any single gene would be different, especially when we only had n equals 10 in this cohort. So we thought, is there some way to normalize the data to magnify the differences between them so that we could self-normalize and maybe find something? So what we ended up doing was taking all the pro-inflammatory markers, which are important at early stages of healing, and just adding them together, and then dividing them by all the reparative markers that are important at later stages of healing, and then making a ratio. So that in healthy wound healing, this ratio is higher immediately after injury, and then it decreases over time. So this is what you would expect in healthy wounds. In diabetic wounds, this caused a 100% accurate separation of healing and non-healing ulcers, in that in this initial 10 cohorts, all of the initial patients who ended up healing, the score went down over time, and everybody who didn't heal, the score went up over time. So that if you used the fold change in that score by 4 weeks, it was 100 times higher for non-healing compared to healing. So not even close, so far away. And now we have, through work with the Diabetic Foot Consortium, we've been able to validate this in a total of 51 subjects across something like 7 sites. And it's still very accurate for predicting healing outcome, which is really useful because if this could tell you, I forgot to mention this is using debrided wound tissue, so it was designed to not add any time to the clinic visits because I know that's very challenging to take care of tons of patients. We're in the process of validating this in another 100 patients through the Diabetic Foot Consortium. And I will say preliminary data does not look as good as this. And I think that's because the patient population is a lot less healthy. They're a lot sicker than in the preliminary cohort. And so that's something that has come up a lot today is the comorbidities and how that changes things. So I think in the breakout session, this is going to be an important part that I would like to discuss. But just one more thing. The problem with that biomarker is that it uses 2 samples collected over 4 weeks, which is not terrible for this patient population, but I think it would be better if you could just have 1 sample. So the patient comes in, you take a sample, and you say, okay, your risk of non-healing is very high, and then fast track them sooner to advanced therapeutics. So there we took a more unbiased discovery-based approach to finding biomarkers. So we took 27 subjects. This was from Alicia Orapalo's group at Northwell. And then we waited to see who healed and who did not heal because no one knows at the time of sample collection. And then we could classify the samples objectively. Then we assayed 275 genes related to macrophage phenotype and wound healing. And then, of course, we used AI, but it was very simple AI machine learning that it's not your black box AI, but you actually know how it works to select the genes that were most predictive or not of healing. Long story short, not a single gene was predictive of wound healing outcome. But we kind of had expected that a bit because that's sort of what we had found before. And so we said, what about ratios? Because the ratio was such a huge game changer in the previous study. So we told the algorithm to check all 2 gene combinations from the 275 genes. And here's something that I could never have an undergrad do, right? So now so this is something, I'm normally an AI hater, sorry, but this was something that was a good use of machine learning to go through and say, what about all 2 gene ratios? And actually all combinations of genes. And we found a lot of 2 gene ratios that could predict healing outcome. This one was C3AR1 divided by CCL22, which happens to be a pro-inflammatory marker divided by a reparative marker. And in the discovery cohort, it was pretty accurate. It got less accurate when we moved to the validation cohort that was a completely independent set of patients. But when you add initial wound area, the size of the wound, it becomes clinically meaningful, 80% accurate. So this is something we're trying to validate now too. Okay, so I wanted to conclude by having 2 questions that I would like to discuss with you all about the 2 areas of research that I just laid out. So the first is, can cell therapies or the ones that I described or maybe others, could they be useful for revascularization in ischemic limbs, since that is how this was invented, was to improve angiogenesis, and what other manufacturing challenges may need to be solved to make that accessible for your patients. And then on the other side, can we discover biomarkers that would predict responsiveness to specific treatments? So instead of saying, okay, this patient is not going to heal in response to the standard of care, well, what would they be good candidates for? Can we use personalized medicine to match them to the treatment that would work best for them? And so with that, I will thank you all for your attention, and sorry for going over time.
Thanks, Kara. A lot of questions, I think, around targeting inflammation. You highlighted that inflammation is required.
Yes.
For wound healing, of course, we know that. So, but a lot this morning we heard about how inflammation was the integrator between all these risk factors in cardiovascular damage and vascular disease. You may not know the answer to this, but what would you, maybe you do, but what would you predict is the net effect of some of the drugs you heard this morning, especially the GLP-1 RAs, on de novo wound healing? Do you think there'd be a benefit? Do you think that attenuating the inflammatory phase could be detrimental? And is there any data?
Yeah, so I think there is data showing that the GLP-1s improve wound healing, but I doubt the mechanism is as simple as inhibiting inflammation, because mechanistic studies have clearly shown that inhibiting inflammation is bad for wound healing, especially venous leg ulcers. And when I was at the Wound Healing Society a couple weeks ago, there must have been 5 or 6 papers showing that increasing inflammation in those wounds was improving wound healing, counterintuitively. I'm positive the GLP-1s are working through modulation of the immune system, especially after learning that the GLP-1 receptor is abundantly expressed on macrophages and T cells, but not on other cells that they regulate. But I bet you it's more of a like a normalization of the way they're supposed to work, as opposed to pro-inflammatory, anti-inflammatory, rather, allowing them to work without those confines of dysfunction.
And through your lens, do you have any ideas about the possible effects of the SGLT2s on wound healing?
I haven't looked into them as much, so I'm not sure.
Okay.
Yeah.
Mark?
I think you raised a really good point that the circulating high sensitivity CRP in the systemic versus what's happening at the wound level could be very different with the GLP-1s. And so I think we don't really know what that interplay is now. I mean, they reduce systemic systemic inflammation, but we're not really sure. I don't know if you've seen what happens to the wound itself.
Well, I just guess I made that comment based on the fact that wound healing is improved, and so I'm marrying that with other studies have shown that inhibiting inflammation doesn't help wound healing. So therefore, I think it's unlikely that the lowering of systemic inflammation is what's causing the improved wound healing. Yeah.
Hey, Dr. Speller. I'm Lauren. I'm a vascular resident, a vascular tissue engineer. Unless it's proprietary, can you share a little bit more information about how you fabricate your microparticles, and if you've tried other polymers like PLLA or PCL or anything like that?
Yeah, great question. So it's proprietary, but I've published many times on it, so you can read it. It's public knowledge now. So we just use PLGA microparticles, which I selected because it's already used in dozens of FDA-approved applications. In fact, I have PLGA in my knee right now from an ACL reconstruction last fall. So it's my favorite polymer for drug delivery. PLLA and PCL are kind of similar to PLGA, so I haven't tried those yet, although I think they would work just the same. And then beyond that, we haven't even scratched the surface. So what I have been focusing on is swapping out the drug, because I'm so curious. With macrophage phenotype being infinite, essentially, I'm very curious how much we can swap out the drug to change the phenotype for different applications. But then the next very interesting thing would be to change the polymer and control the release kinetics. So I haven't even tried that yet. Yeah, thank you.
Other questions out there? I'll ask one more general question, which is sort of the intersection between what you're trying to study and infection, which is obviously potentially at minimum a big confounder, especially in looking at human samples and trying to understand what's the appropriate balance of wound healing in the presence of, it's not a sterile inflammatory environment.
Right.
it's an inflammatory environment that also has bacteria.
Yeah.
So how does your research inform by that tension between inflammation and infection?
Well, I guess I will say I view the microbes in the wound definitely influence macrophage phenotype, just like all the other things that are in the wound. So in our biomarker work, we kind of think of the macrophage markers as like a molecular readout of all the things going wrong in the wound, which may include, a ton of infection, or it may include glycated collagen, which happens with poor glycemic control. So I kind of was agnostic about what's causing the problems within the macrophages, but then using the macrophages as like a readout, right? But I've been collaborating with Lindsay Kalin, a great microbiologist who specializes in wound healing at McMaster University. And so what we have right now is data from 250 patients from the Diabetic Foot Consortium. And we sequenced all the microbes and all the human genes. So we have 250 subjects times terabytes of data for every single one. And the question is exactly that, is how does the microbial communities interact with the human immune cells, or all the cells, because we have all that, and how does that differ in healing and non-healing wounds, and can we stratify patient subpopulations based on that information?
So since you've mentioned it a few times, just for the people in the room who are going to be thinking about projects that might be, asking unique questions about mechanisms of treatment or disease, Diabetic Foot Consortium has been going on now for about 6-7 years. Recruiting subjects is now got over 850 patients who are in the consortium, whose wound healing trajectories have been followed, but who also have a biorepository of blood and tissue samples for these kinds of research studies that are vetted by a committee. So something to keep in mind for people with different questions in their mind about how diabetes and vascular disease might intersect with some of the targets we were looking at this morning. So thank you, Kara, so much.
Yep.
I don't know if Manesh is available, but